Hello everyone and once again welcome to Bioline. In today's lecture we will talk about the operon with the help of an example that is a very famous one, lactose operon. Okay, so let's start about that. So first of all, what exactly is a operon? So I hope you people know that in bacteria what happens, there are the genes which are having the similar function. They are kept under the control of a single promoter, right? And the mechanism via which these all genes are being controlled, that involves a system of operon. Okay, so the operon system was actually, this concept of operon was given by Jacob and Monod. ठीक है, Jacob and Monod ने ये concept दिया था. What exactly a operon is? It is just a cluster of genes where we have the structural genes, we have the promoter, we have an operator, we have a regulatory genes. Okay, so the collection of all these genes together is called a operon. So we'll be talking about the famous type of operon, that is the Lac Operon. The Lac Operon is also known as, here the word lac stands for lactose. That means this is the operon which is we can say, which is concerned about the metabolism of the lactose sugar. Okay, it is related to metabolism of lactose sugar. Okay, now, so first of all, there are two important properties that we need to remember about the Lac Operon. That is, it is an inducible operon, and second, that is a negatively controlled operon. Okay, so first of all, let us see what is a inducible operon. Inducible operons are the operons which are normally off. That means with the transcription of the genes which are there in this operon are normally off. They are not needed by the cell. That's why the cell will not unnecessarily make the product of those genes and waste its energy. Okay, so these genes will be only switched off when they are required. So this type of operons which are generally consist of the genes which are generally normally off. Okay, that means operons is the transcription translation is not taking place, and there has to be a stimulus. That means something must happen to induce the transcription or turn it on. Okay, we can say here in the normal cases what will happen, the operon is off. So there has to be a stimulus. Unless and until there is a signal or there is a stimulus or there is something is there, will be there which will convert this off into the on mode. So this has to be induced. The operon has to be induced. The transcription translation of these genes which are there in these operons has to be induced. That's why this operon is called inducible operon. Okay, after this is also called negatively controlled operon. Why negatively controlled? Because this is controlled by a regulatory protein which is known as repressor. Repressor means which is going to have the negative effect, or it is going to suppress the activity of the genes which are there in this operon. Okay, so what we will say, this is a negatively controlled operon because the repressor will bind to the DNA and inhibit the transcription of the structural genes which are involved in this operon. So why this, I hope it is clear that what is an inducible operon and what is a negative operon, and Lac Operon is inducible and negative operon. Okay, let's move further. Okay, upstream to these structural gene we have a sequence which is known as operator region. Okay, operator region is the site where actually the repressor is going to come and bind. Always remember that this is the position, or we can say the site on the DNA where the repressor will be binding. Upstream to that there is a promoter, and this promoter is for all these structural genes. So this is called P-LAC. That means promoter for the genes which are responsible for lactose metabolism. Okay, upstream to that we have a regulatory gene that codes for the repressor protein. It is represented by small I, and then we have a promoter for this regulatory gene. So all these are the regulatory sequence, and from here that is a promoter. From the promoter for the Lac genes to the end of the Lac genes is this entire is called the Lac Operon. So this is the general structure of a Lac Operon. We'll move further. Okay, in this figure it is also the Lac Operon is given. You can see here this is the gene Z, this is Y, and this is A. Okay, this is the operator region, this is the promoter for these structural genes, this is the regulatory gene, and this is the promoter for the regulatory genes. Okay, so see, lactose is the sugar which has to be actively transported within the, it has to be actively transport within the bacteria. ठीक है, बैक्टीरिया के अंदर इसे एक्टिव ट्रांसपोर्टेशन के थ्रू लेके जाना है, एंड देन ओनली द मेटाबॉलिज्म ऑफ द लैक्टोज कैन बी कैरीड. Okay, and for the transportation and even for the metabolism, the product of the three genes, that is Z, Y, A, are needed. The product of Y is Z is beta galactosidase. Okay, and what it will do, it will help in the cleavage of this disaccharide into the monosaccharide, that is galactose and glucose. Okay, so beta galactosidase will break the lactose into galactose and glucose, and then it will be utilized directly in the glycolysis pathway. Okay, it is utilized via the glycolysis and all. The next function of the next gene, that is the Y gene, is that the Y gene codes for permease. We can say the galactosidase permease. It is also known as lactose permease. What is the function of permease? Permease protein is going to act like a channel. Okay, and it will locate itself on the plasma membrane of the bacteria. Okay, this is a permease protein, and this channel will allow the passage of lactose from the media into the cell, like this. ठीक है, ये उसके passage allow करेगा. Next, the third, that is the A gene codes for thiogalactosidase transacetylase. It will help the cell to get rid of the toxic thiogalactosidase that also get transported by Lac Y. Via this, some toxic materials also enters. Okay, so what this transacetylase will do, this will transfer the acetyl group on these toxic materials and will get rid of the toxic compounds. Right, let us move further. You can see here, this is what the diagram look like. See here, this is the cell membrane of the bacteria, and this is the permease. Permease is the product of gene Y. Okay, so what is happening? Permease is acting like a channel here, and this is the disaccharide extracellular lactose. That is, the lactose is present in the media. So what will happen? This permease will form a channel here. Okay, and then this lactose will enter here. Once the lactose is within the cytoplasm, the enzyme beta galactosidase is going to act on it. Beta galactosidase will also perform a very important function here, that it will convert the lactose into a related compound known as allolactose. ठीक है, ये lactose को allolactose में convert कर देगा. Okay, also the beta galactosidase will act on both the lactose and the allolactose and break that disaccharide into the monosaccharide galactosidase and glucose. Okay, what is happening? Beta galactosidase is performing two functions. Number one, it is converting some of the lactose into the another related compound, the allolactose, and the another function that will break both the lactose and the allolactose to the galactose and glucose, and these are the monosaccharide which is easily utilized by the bacteria. ठीक है, इसको आगे इसका utilization हो जाएगा via glycolysis. Okay, now see here. This lactose operon is generally, I hope everyone you are able to understand now, this is the lactose operon, and it is coding for the genes which are responsible for lactose metabolism. Right? और मैंने अभी आपको बोला कि during the synthesis of a protein, a huge amount of energy is required. So the cell will always want to control the expression of the gene. That means whenever the product of the genes will be needed, only at that time the genes will be transcribed or translated. Okay, otherwise then what is the need to unnecessarily waste the energy? Right? So whenever, remember, when the lactose will be there in the medium, then only its metabolism has to be done. Yes, जब lactose medium में होगा तभी उसका metabolism cell करेगा, तभी cell को जरूरत है कि वो permease बनाए और lactose को अंदर लेके आए. Right? और तभी उसको जरूरत है कि वो beta galactosidase या transacetylase बनाए. So that's why this lactose operon is only operon will be switched on in the presence of lactose. Right? If there is a lactose, then only the operon is switched on. Okay, if there is the absence of lactose, if there is absence of lactose in the media, if the media do not have the lactose, then what will happen? The operon is set to be switched off. Operon क्या रहेगा? बंद रहेगा. Genes क्या रहेंगी? Activate नहीं होंगी. Why? When there is no lactose, there has to be no metabolism. So unnecessary energy का wastage नहीं होगा. Right? Okay. Now, You can ask a question here कि when हमने क्या बोला था यहाँ पे, okay? चलिए, we'll talk about this later. First, we'll try to understand the regulation of the Lac Operon. Now, Lac Operon is regulated via two proteins. One is an activator, which is known as CAP. Okay, I'll write the full form, that is catabolite activator protein, या activation protein. This is a positive regulator. Why? Because this cap is actually an activator. So यहाँ पे this is an example of what? This is an example of positive regulation of Lac Operon. The another important protein which involved in the regulation of the Lac Operon is the repressor, जिसे कहा जाता है Lac repressor. This is a repressor, that means it is going to be negatively controlling the operon. So Lac repressor, the name itself is suggesting that it is going to negatively control the operon, and activator cap is catabolite activator protein. It is going to positively regulate the activator, the Lac Operon. Okay, so first let us try to understand this. Okay, first we'll try to understand this. We'll try to understand the control by Lac repressor. This is an example of which type of control? A negative control. Okay, this is a negative control example. Now, as we have seen that the Lac repressor is coded by the Lac I gene, which is a regulatory gene. Okay, and it is located near the other Lac gene. Okay, but transcribed from its own constitutively expressed promoter, which you can see here. If I'll draw this, this is the Lac Operon. This is the promoter for the Lac I. Okay, and this is the Lac I gene. This is the Lac I gene. Okay, then it is also near to the promoter for Lac genes, and this is the operator region, and this is the Z, this is Y, and this is A. So you can see here the Lac repressor is actually coded by Lac I gene, which is there in the, we can say, which is nearby located to the other Lac genes. Okay, but this Lac I is having its different promoter. Okay, इसका promoter अलग है. This is its promoter of Lac I, and the promoter of other Lac gene, Lac ZYA, is Lac promoter. This one is the promoter for Z and Y and A. Okay. Now you can see here, यहाँ भी हमने बात की, this is the promoter, this red one is the promoter for gene ZYA, and this orange one is the promoter for the regulatory gene, that is I, which is coding for what? Which is coding for the repressor protein. So इसका promoter ये है, and this is the promoter for the ZYA. The promoter is different, उसका promoter अलग. Okay, now याद रखिए the Lac repressor, नाम क्या है? Repressor. That means it is going to negatively रेगुलेट द ओपेरॉन. मतलब ये ओपेरॉन को क्या करने वाला है? याद रखिए, ऑफ करने वाला है, सप्रेस करेगा. तो आपको सप्रेस करने की जरूरत कब पड़ेगी? व्हेन देयर इज नो लैक्टोज. ओके, सो दिस लैक रिप्रेसर कैन बाइंड टू द डीएनए एंड रिप्रेस द ट्रांसक्रिप्शन ओनली इन द एब्सेंस ऑफ लैक्टोज. जैसे मैंने अभी आपको बोला था, इफ देयर इज नो लैक्टोज, देयर इज नो नीड फॉर द लैक जीन. दैट्स व्हाई व्हाट विल हैपन? द रिप्रेसर विल गो एंड बाइंड वेयर इट इज गोइंग टू बाइंड टू द ऑपरेटर region, okay, which is just upstream to the structural genes. If there is lactose, agar lactose hoga, then repressor will be unable to bind. Okay, this will be unable to bind. ये operator से bind नहीं कर पाएगा. Okay, in the presence of lactose sugar, the repressor has to be inactivated. Repressor क्या हो जाएगा? Inactivate होगा, and the genes are de-repressed, मतलब the genes are expressed. How does it happen? Let us try to understand. Okay, first we'll consider this situation. This is the Lac Operon, which you can see the gene Z, this is the gene Y, this is the gene A. This yellow one is the operator region. Sorry, this is the promoter region, and an operator region is actually, it is overlapping on the promoter and the Lac Z region. ठीक है, उसके ऊपर overlap करता है, which you can see here. It is not a separate one जैसा हमने पहले देखा है, but it is just overlapping. It consists of some of the sequences of promoter and some of the sequence of the Z gene. Okay, so this much is the operator, which I have shaded with the red one. Okay, you can see here, this is the regulatory gene Lac I, which is going to produce, or which is going to synthesize what? It will be synthesizing the repressor, and this is the promoter. So the condition is when there is no lactose. Generally, if we just talk about what should be the situation in the Lac Operon should be off. Why? Because there is no lactose, right? Lactose off है तो यहाँ पे operon क्या होना चाहिए? बंद होना चाहिए. And how can the operon be off when? Because here the lac repressor must be active. In this case lac repressor क्या होगा? Active होगा और जाके operator पे bind कर रहा होगा. Okay, let us try to understand. First I just delete it from here. Okay. Now see here, what is happening when the lactose is not there? The promoter will give the signals to the structural gene and it will initiate the transcription of repressor. The transcription and translation will be there. There will be active lac repressor. This lac repressor will go and bind where it is going to bind to the operator region. This much is the operator region, right? This is all is the operator region. You can see here it is binding to the lac O, that is operator region. As a result, now what happened when RNA polymerase will go and bind to the promoter, it is not able to move further. यहाँ पे क्या हो जाएगा? ये RNA polymerase, if I'll just change the color of RNA polymerase, if this one is the RNA pol, it is unable to move further. Why? Because this repressor which is bound to the operator, it is creating a hindrance. ये यहाँ पे एक restriction जैसा काम कर रहा है, ये उसको आगे बढ़ने नहीं दे रहा. As a result, there is no transcription or translation for this particular gene and there is no transcription and we will say the operon is said to be switched off. Operon क्या हो गया है? Switch off हो चुका है. Why? Because यहाँ पे lactose नहीं था and already the repressor is in the active mode. The repressor will go and bind to the operator. As a result, the RNA polymerase which bind to the promoter, it will not be able to move further and it will not be able to move to the structural genes. Right? We'll move. Now we'll see the situation when the lactose is present in the media. Okay, so what should be happening here? Because the lactose is present in the media, that means the lac genes, that is the product of the ZYA, will be needed here. Why? Because the genes are required, the product of the genes are required for the metabolism. So the operon has to be switched on here. Okay, and how can the operon be switched on? When the repressor has to be inactivated. That means here something must happen to inactivate the repressor. Repressor कैसा होना चाहिए? Inactive होना चाहिए. Right? So let us understand what happens here. Here, what will happen? A very small, as soon as the lactose is present in the media, a very small amount of lactose will start entering into the bacterial cell. Okay, entering into the bacterial cell. Once the lactose, the small amount of lactose, this will be initially, it will be very small amount. Okay, initial lactose which will enter into the cell, okay, will be very small amount. This small amount of lactose will be converted into allolactose via beta-galactosidase. I told you that beta-galactosidase is having two functions. What? Number one, it can break the glucose, the lactose into the glucose and galactose. The second important function is what it will do is that this lactose will convert beta galactosidase will convert the lactose into allolactose, and this allolactose is going to act like a inducer here. Inducer means this is going to induce this operon to get on. ठीक है, इस operon को on कौन करवाएगा? यही inducer करवाएगा, which is the allolactose. Always remember, if you don't have the option of allolactose in the question, you can tick lactose is the inducer in the Lac Operon. But if you have the option of allolactose, always go to the allolactose. So allolactose is the inducer. What this allolactose will do? See here. When the lactose is present in the media, even at that time, the regulatory gene, this lac I is our regulatory gene. Lac I. This is producing an active repressor. The repressor is in the active form here. Okay, but now because allolactose is there in the media, the allolactose is within the cell. This allolactose will go and bind to the repressor, which you can see here. As a result, there will be the conformational change in the repressor, which will inactivate the repressor. The repressor is now inactivated, and because of the conformational change, it will not be able to go and bind to the operator region. Also, this allolactose will go and bind to the repressor which is already bound to the operator. Okay, if there is a repressor here which is bound to the operator, this allolactose will go and bind here, and again it will call the conformational change, and because of that change, the repressor will leave the operator and it will come out like that. Okay, so via both the mechanism that allolactose will bound to the free repressor and inactivate it, and also it will go and bind to the already bound repressor, and also it is going to inactivate it. And as a result, what happened, the repressor is now unable to bind the operator and the repressor is inactive, which we needed. And then again here the RNA polymerase will come and bind to the promoter region, and now it is free to move because there is no hindrance here. It can easily scans all the structural gene. There will be the transcription of the gene followed by the translation, and there will be huge amount of beta galactosidase enzyme, permease enzyme, and the transacetylase enzyme in the cell. Okay, and then what will happen, a large amount of glucose will enter into the cell from the meat. Okay, everyone. So this is the situation where the lactose is present in the media. We'll just revise it. When the lactose is present in the media, there will be definitely an active regulatory protein will be there. That means an active repressor will be prepared. Repressor के preparation में या production में कोई फर्क नहीं पड़ेगा. It will be same. Okay. What happened? A small amount of lactose will enter into the cell. Okay, and it will convert into allolactose, which is our inducer. This inducer will bind to the free as well as the bound repressor and cause the conformational change in the repressor, and it will inactivate the repressor. Now the repressor will lose the ability to go and bind to the operator region. Now RNA polymerase can easily bind to the promoter and it will have no hindrance. It can easily transcribe all the three structural genes, that is ZYA, and it will be followed by the translation, and we will get the enzymes in a huge amount, that is galactosidase, permease, transacetylase within the cell. Okay. Now you can have one question here. I have told you in the previous slides that when there is no lactose, जब lactose नहीं होता, the operon is said to be switched off. That means there is no beta galactosidase, no permease, and no transacetylase production. उनका production नहीं हो रहा है. Then how come this small amount of lactose, जो हम बात कर रहे हैं कि once the lactose is present in the media, the small amount of lactose will enter into the cell. How come this lactose is entering into the cell? Because we don't, other lac genes are not in the they are not active, they are not transcribing, right? So यहाँ पे permease कैसे आएगा? The reason here is within the leaky, or we can say basal level of transcription. Okay, always remember here the repression by the lac repressor is not very tight repressor. Okay, some amount, a very minute amount, that is a leaky or a very small basic level of transcription of these lac genes is on even in the absence of the lactose. ठीक है, तो याद रखिएगा when lactose is not there, even at that time a very small amount of beta galactosidase, permease, and transacetylase will be transcribed within the cell. Okay, and this type of transcription is called leaky transcription. Why? Because lac repressor is there. Okay, it is repressing the operon, but the repression is not very tight repression. That's why some leakage will be there. Some basic level of transcription of these three genes will be there in the cell, and that's why always in the cell there will be some small amount of permease, small amount of beta galactosidase, and small amount of transacetylase. So as soon as lactose comes in the medium, in the surrounding, there is already some permease, a small amount of permease will be there on the cell membrane. ये यहाँ पे कुछ permease already present होगा, and that's why this small amount of lactose can enter into the cell. Once it enter into the cell, because the cell also have small amount of beta galactosidase, because of this leaky transcription, it will convert into the allolactose, which is our inducer, and it will induce. What it is going to do? It will remove the repression by what? By the repressor. ये repressor को inactivate कर देगा, and then the transcription now will go with a very very high rate. The transcription will increase with a very higher rate. Okay, so this is all about the Lac Operon. We have already talked in the previous slide that the Lac Operon is regulated by two proteins. One is our lac repressor that we have already seen. The another protein, which is the cap protein, which is also we can say the positive regulation of Lac Operon, that we'll see in the next video. That will be the Lac Operon part 2 video. Okay, so all of you will meet in the next class. Just okay, have a good day.